thanks everybody for coming. And John, you have precisely 15 minutes, please. And I will skip an introduction. Everybody knows you, so have fun with your talk. We are looking forward to it. You need to...
yes, I have to remember how to unmute myself when the thing comes up. Thank you, Nikolaus. Thank you to the other organizers for putting together. I really enjoyed that first session, really outstanding talks, and it's such an exciting area to work in. It's a pleasure to be here today. I'm always a bit alarmed when you're introduced with everybody knows who you are, because that can hide a multitude of sins. But anyway, so what I'd like to talk to you about today is an application of some methods for computational strategies for spatial transcriptomics, and in particular in the context of early mammalian development. We've heard in the previous talks that the mouse brain and the developing mouse brain is a really nice system to study. We're going a little bit earlier than that. We're going to look in early gastrulation and early organogenesis. So in the mouse, this is between embryonic day 6.5 to embryonic day...
John?
Oh. Yeah, John, you should swap your displays if you want, so that we...
Yep.
Please.
Of course. I'm never quite sure what it's going to do. Okay, you should see the right one now, yes?
Yes,
perfect. Okay, thank you. So yeah, so I was saying embryonic day 6.5 through to around embryonic day 9 and just after. And of course, there's a great heterogeneity of cell types that are present in the embryo at these stages of development. So it's particularly amenable to single cell approaches. And indeed, over the last 5-6 years or so, together, mainly with colleagues in Cambridge, we've been applying what I suppose we can now consider classical single cell genomic strategies, in particular, single cell RNA sequencing, to look at the different cell types that are present. Here's some work for the lab of Bertie Gutkins, where we looked at 9 sequential time points from E6.5 to E8.5, and these UMAP plots that we're all familiar with for how we go from early cell types in the epiblast through here towards the blood, the endoderm at the bottom, various neural and mesodermal cell types in the middle. And with Wolf Wright's lab, we've been looking at multi-omic strategies. A number of the previous speakers mentioned that how important and how useful it will be to have these within a spatial context. And I absolutely agree, because with having this chromatin accessibility, DNA methylation, and RNA expression information from the same cell with Wolf's lab, we were able to understand something about how the hierarchies of different cell types or germ layers are established in early development, with ectoderm-specific enhancers being marked relatively early on and not undergoing the same repression as endoderm and mesoderm-specific enhancers, which are in turn activated later on in development. But of course, and the purpose of this workshop is that whilst powerful, single-cell genomics approaches are limited in that they lack spatial context. And at these stages of development, there do exist in situ databases, but they're limited by the lack of a common scaffold and typically by the inability to measure expression at single cell resolution. And at the time points that we're looking at here, the cells are typically very compactly located. So you really do need that single cell resolution in order to discriminate, tease apart what's going on between cells, especially when there's so much heterogeneity in such a relatively small number of cells. And so we teamed up with the lab of Long Kai, and Long will be talking next. So I'm not going to dwell too much on the technology that his group have pioneered over the last 5 to 10 years, but suffice it to say that they use a highly multiplexed RNA FISH approach in order to study the expression of now up to hundreds of genes, or indeed thousands, within an individual cell. And so we teamed up with Long's lab to design a library targeting 351 genes that are cell type specific during early development. And then we used his seq-FISH technology in order to probe these. And we've done this now at multiple time points. But what I'm going to talk to you about today is data from the latest time point that we were interested in, which is around embryonic day 8.5. And this is the 8 to 12 somite stage. So what I'm showing on the left are cartoons here of the embryo at this stage of development. And what Tim Lohoff, the experimentalist, did here was he cut sagittal sections through the embryo, so you can see them indicated here. And he did this for 3 independent embryos. And these sections are shown on the right-hand side here. And then having cut these sections and embedded them appropriately, they were subject to seq-FISH technology in Long's lab in California. So in terms of the analysis, of course, this is a complicated and challenging process from decoding spots through to the much more downstream analysis. In the work we did here, we used Elastic. This is a tool developed by my EMBL colleague Anna Kreshuk and EMBL Heidelberg, which is a machine learning based strategy in order to do the segmentation. And we were able to take advantage of a particular tweak to the experimental protocol where we particularly targeted for proteins that could be used to stain the cell membrane, and that really helped facilitate the segmentation. And this can be, especially when you have compacted cells, a really challenging problem. So an example of the segmentation is shown here. So you can see this is the markers that were used to stain the membranes. This is the segmentation. So this is one particular segmentation mask. And then within each of these little turquoise areas, you can count the number of RNA molecules that are expressed from each gene, and that gives you, in the context of single-cell RNA sequencing data, an expression matrix where each cell is, of course, now indexed by its spatial location. The data are of really quite nice quality, and we can ascertain this by looking at the expression of known marker genes. So what we're plotting here are effectively, you could consider 12 digital in situs for 12 different genes that were probed in the experiment, from genes that are associated with the heart, such as titin, through to genes associated with an endodermal identity, FOXA1, Claudin4, and they are indeed expressed in the regions of the embryo where you would hope them to be, giving us some degree of confidence that the experiment has worked. But of course, the real power of this experiment is the fact that we're asking all of these different genes in parallel. And so the first step from a computational perspective is to take each of these segmented cells that we've obtained and to assign them a cell type identity. And now one could do this by just clustering the seqFISH data on its own, but actually you're looking at 351 genes. So you can argue that the granularity of cell types that you're going to be able to identify might be slightly limited. And so we reasoned that we could integrate these data with our single cell RNA sequencing data to get a much more refined clustering. And in order to do this, we used an approach that we'd put out now a few years ago by my then postdoc Lali Hagverdi, who's now a group leader at BIMSB in Berlin, so Nikolaus will know her well, for integrating multiple single-cell RNA sequencing data sets. And this approach effectively is motivated by the idea of finding pairs of mutually nearest neighbors between cell types that are related in a high-dimensional space. So for each cell type, we try, or for each cell in one experiment, we'll try and find its closest neighbors in a second experiment, and having identified those, we'll use these effectively to map these two different experiments on top of one another so that we can collapse the manifolds on top of one another, and then we can successively integrate more and more data sets. And this, together with a paper that came out back-to-back from Raul Satish's lab, has, I guess, set up a little cottage industry of approaches for batch or data set integration for single-cell RNA-seq. But what we can do is we could take exactly this approach and apply it to our seqFISH and single-cell RNA sequencing data. So what we do is we take the 350 genes that we probed with seqFISH, and we subset the single-cell RNA sequencing data from the same developmental time point to look at those 351 genes, and then we effectively map the seqFISH dataset onto the single-cell RNA sequencing dataset, and then via transfer learning, we look at the cells, the neighboring cells in the single-cell RNA-seq dataset to which each seqFISH cell is mapped, and use that to assign it a cell type identity. And so we can then visualize this, of course, in space. So this is now one embryo, and I've separated it by germ layer. And what you can see is that each cell is now colored by the cell type to which it's assigned. So we've got ectodermal cells here on the left, mesodermal cells with a purple heart population in the middle, endoderm cells corresponding to the gut tube down here, and a small number of extra embryonic cells. And you can already begin to discriminate between spatially coherent and diffuse cell types, and you can indeed quantify this more statistically by examining whether there's any residual gradient patterns in space after you've assigned cell type identity. So this is nice, but actually we can get a lot more from this data, of course, than just pretty spatial clustering. And one of the things that we were very interested in is, could we go from this 351 gene profile to a 10,000 plex in situ? And so what we did here was, again, using these ideas of mutual nearest neighbors to impute the expression of genes that weren't profiled in seqFISH. So we map each seqFISH cell, spatially resolved, to single-cell RNA sequencing data, and then we find its neighboring cells in single-cell RNA-seq. And then for each gene, we take its average expression across those cells and use that as its imputed expression profile. And what I'm showing here is a validation of this imputation strategy. So this is now 6 genes that we did not profile using seqFISH, but we did profile using classical single molecule RNA FISH. And at the bottom here is their imputed expression profile. And at the top is the independent orthogonal validation. And you can see that both for specific genes, as well as broadly expressed genes and negative controls, we seem to be doing a pretty good job. And this suggests that our imputation strategy is both specific and sensitive, and that allows you now in space to probe the expression of up to 10,000 genes within their very precise spatial context. And just to close, one particularly nice biological application of these data comes in looking at the development of the gut tube. So beautiful work from Kata Jantanakis and Dana Pear's lab in Memorial Sloan Kettering has probed the expression of genes along the anterior posterior axis of the gut tube. And many of the genes that they characterized as being expressed in particular locations were profiled using our seqFISH experiment. Here they are boxed in red. And so we could use this set of genes to further refine the annotation of cells along the gut tube, which in our previous experiment had all been just assigned an endodermal identity. So this is that overlay of Kat and Dana's data together with the seqFISH data. And you can now see that we're able to assign each of these endodermal cells a very precise identity on the basis of the downstream cell population that they're likely to contribute to. We can plot this in space. So this is now for 2 embryos, embryo 1 and embryo 2. And if we zoom in a little bit on the gut tube, you can see that cells along the gut tube are colored by cell type. And indeed, this forms a very nice anterior posterior pattern. So we've got, for example, lung 1 cells here on the ventral side and lung 2 cells on the dorsal side. But why are these lung 1 and lung 2 cells not assigned different identities? Well, they couldn't tease them apart on the basis of the single cell RNA-seq in the original paper. But what we could do with the seqFISH is look for differentially expressed genes. And indeed, we find markers of genes that are associated with either the dorsal or the ventral side of the gut tube. And they're just plotted here. And then we could validate this. And we did this together with Kat's lab at Sloan Kettering using HCRs. So we can really validate that these genes are indeed dorsally and ventrally expressed. And then perhaps most excitingly, when we look at their identities, we can see that the lung 1 cells, the ventral cells, are likely to, in terms of the genes that are specific, likely to correspond to cells that have a tracheal or lung identity downstream. So they are progenitors of trachea and the lung, whereas the dorsal cells are esophagus progenitors. Now, this dorsal ventral separation of these cell types was known, but it wasn't known to arise until about 24 hours later in development. So we can see that at the RNA level, this segregation seems to be pretty early on as development proceeds. So in terms of what I see as the outlook, so prefacing Fabian's question before I even get a chance to answer it, I think we've shown how you can combine multiple different data types to quantify molecular patterns in space. I think there's really interesting opportunities to think about how we integrate this with nuclear tracking data. So beautiful work from Philip Keller's lab in Janelia profiles how cells migrate or nuclei migrate throughout this 48 hours of development, overlaying our data with this data from the Keller lab and also Katie McDowell, who has her lab now at LMB, is going to be very powerful, I think. But I think the real challenge maybe for the field is going to be moving beyond these statistical descriptions of data to more fundamental understanding. So can we integrate some of these statistical correlations, which are of course very important, with a more mechanistic understanding of why cells actually make the decisions they do? And I think that we are now getting to the point with spatial, temporally resolved data at multiple molecular modalities where we can begin to ask some of these challenging and big questions. And I think that's tremendously exciting because it allows us to move from descriptive models to really a deeper mechanistic understanding. And with that, I'll thank my wonderful collaborators. I'm lucky to have a great lab. I enjoy working with all of them. On the work that I presented today, that was especially collaborations with Wolf, Bertie, and Long, as well as Jenny Nichols. And I think that I've listed the people on slides who contributed. So with that, I'll close and I look forward to the other 2 talks in this session and to answering any questions that anyone might have at the end of that. So thank you very much.
John, this was truly fantastic. I can hear the applause. Wonderful. And you're actually 1 minute faster than scheduled. So I'll take the liberty to ask you just one question. So if you say you want to get at a more fundamental understanding of the processes during development using these kinds of data, you will also need to perturb somehow the system to be able to really match up with predictions and reality. So how do you intend to go about this?
It's a great question. I think that, so previously we've used chimeras where we've injected mutant cells where a particular gene is knocked out and they're labeled, they're injected at the blastocyst stage, and then you can ask whether they contribute differentially towards particular lineages. I think that's an interesting challenge. I think a secondary challenge though, and something that we didn't really face in single cell RNA-seq is, how are you going to map morphological differences onto a reference? Because a lot of the perturbations you might consider could be quite drastic in terms of the morphological changes. And if it's too drastic, then somehow mapping those changes onto a spatial reference is going to be rather difficult. So I think we have to think quite carefully about the sorts of perturbation experiments we're going to perform. I think in some sense, we're not going to so much want to take the wheel off a car. We're going to want to maybe just change slightly the rate at which you can brake. Yeah, because otherwise it's going to be impossible. So I think brute force perturbations are not maybe the way forward. So thinking intelligently about that is going to be hard, but really important.
Yeah, so thanks very much for this inspired answer. We need to move on. Thanks again, John. And you introduced already the next speaker, Long Kai, basically. So I also don't need to do this, but just to say that we are very happy that you could make it along and present your latest work as a pioneer in the field. Thank you.
Well, thank you, Nikolaus, and thank you, Fabian, for the invitation. I'm very happy to be here. And I want to tell you about our most recent work in looking at nuclear structure. So as you know, we'll all see in textbooks that DNA is organized in this hierarchical fashion, in double-stranded DNA, and then packaged into nucleosomes, and then form chromosome fibers. But then, on the other hand, if we take a microscope image, this is the middle panels we usually see is some kind of dappy image with bright spots and dim spots inside nuclei. So how do we reconcile these two pictures, and how do the chromosomes actually look like in single cells? And so what we know so far is a lot of it is based on genomics, where you use different methods to analyze the interactions between DNA locus, and this is by Hi-C, GAM, or Sprite, and you can generate these contact maps, generate these like block-like structures, and from that you can computationally reconstruct what the chromosome is supposed to look like based on constrained by these contact maps. And so we learned quite a bit from these measurements, but on the other hand, this is a reconstruction. We can also directly image the cell and look at the DNA locus using DNA fish or chromosome paint to get a sense of what the gross chromosome structures look like in individual cells. But imaging methods typically are hard to scale up, to the genomic level. So as John mentioned already, we developed this method called seq-fish that allow us to multiplex a very large number of molecules in cells, and it uses multiple rounds of hybridizations to encode different transcripts for different molecules with different sequence of colors. And this way you can scale up very quickly, even if you have only four fluorophores available in microscopy. And we developed this method a number of years ago, and there are other methods that uses different coding schemes, but uses roughly similar ideas. And so we showed a couple of years ago that you can actually cover on the level of the transcriptome using this method. We call it RNA seq-fish plus, where you can go into tissues, you can zoom in successively into first one part of the brain, the cortex, and zoom in further into a cell, and then zoom in again in this almost diffraction limit region. And each grid here is about 100 nanometers. You can see in super resolution the transcripts inside the cell, and then use this transcriptome information to cluster the cell types and so on. And one of the advantages, as John mentioned, is you can look at the spatial patterns of gene expression, the cell types, but also the cell-cell interactions, because you can now look at the signal molecules and the receptors within this native context of tissues. So, for our most recent work, we integrated this method, RNA-seq-FISH, as well with DNA-seq-FISH, and which allow us to look at thousands of regions. So we looked at about 2,000 regions that are roughly 1 megabase of resolution apart in the whole genome, and then another 1,000 regions about 25 kilobases. So on top of these measurements, the RNA and DNA measurements, we can also look at multiple antibodies, immunofluorescence, and there are about 17 antibodies. So this is what it actually looks like. Here's a single cell reconstructed, and then we're just going through different chromosomes. All the dots are basically all the thousands of loci they're detected in the cell. And you can see in many cases, the chromosomes are separate to alleles. Sometimes they're fused together. This seems to be pretty random how that happens. And after that's done, now we can look at the antibody marks. So these are the heterochromatic marks that mark the repressed regions. And then it's the nucleolus. And then now we're going through the active marks, the RNA polymerases, the speckles, and then finally the lamins and the repeat regions. So you can get all this information integrated into a single cell. And that's a nice thing about actually using these imaging approaches, because you can just continue to do these experiments on the same tissue. It's very straightforward to do intracellular as well as extracellular proteins, all as well as RNA, all in the same sample. Okay, so if you look at this chromosome structure, you can see if you zoom into the chromosome 5, there's 2 copies of it, and they form this kind of wiggly structure. And you zoom in further with the 25 kilobases on just one part of the chromosome 5. You can see further, at even higher resolution, what the chromosome shape looks like. And, you can stare at these images for a very long time. And that's what I've done during the pandemic, is to look at they're very beautiful structures to look at. But ultimately, there's very little actually can say about them, besides they're squiggly and then they're very heterogeneous in single cells. This is to show the validation that you compare the DNA-seq-FISH plus measurements versus high-C measurements. You get very good agreements on different scales, all the way from chromosomes to megabase to kilobases. And then so now, with this technology, right, you can now take the same DAPI images and overlay essentially this high-resolution chromosome structure on top of it. So as I was saying, the structure itself is very heterogeneous in single cells, and what really makes sense is if you superimpose the immunofluorescence on top of this DNA image. So in this case, what we did was use a oligotagged antibody, and then just and there's 17 antibodies dumped in at the same time, and then we can use FISH to basically read out these oligotags on antibodies. So we can iterate through all these different antibodies. You can see in the same cells, and then compiling them together, you can see the heterochromatic regions, the active regions, the nuclear lamin, the repeat elements. So, for example, chromosome X has a lot of line elements. So you see the X dots and the line elements localized together, and then finally the nucleolus and then the RDA carrying chromosomes are overlapped on top of each other as well. So if you take the DNA locus, right, overlaid on top of these chromatin marks, what you can do is you can measure how often a DNA locus is close to a particular chromatin mark that you see in the immunofluorescence. And you can generate maps like this, where going across the chromosome, you can see the frequency of those loci being near, in this case, about 300 nanometers, which is diffraction limited, to this particular acylation mark. And what's surprising is that this actually matches the ChIP-seq results for H3K9 acylation very well. And I should mention this is at a megabase resolution, of course, because that's the resolution of this data. But it's surprising that they match at all. And what's further interesting is that if you look at single cells, so each row is a single cell here, a lot of these features are actually fairly deterministic, even though the chromosome structure itself looks very random. And this is true not just for this particular mark, but actually true for many marks. And just another example showing that the nuclear speckles, again, it matches the sprite measurements for these speckle compartments. And then again, it's also very deterministic in single cells that these locus is always interacting strongly with the speckles, even in single cells. And so we call, and some of these you can see are quite, close to 1 in terms of the frequency of interaction. So almost all of the locus in single cells are interacting. And we call these fixed locus because they're kind of fixed. And so what do they actually look like in single cells? Because we have the images, right? We can go back and look. So in purple shown, the light purple is the actual nuclear speckles, and the purple dots are these fixed locus. And you can see that they're actually coated on the surface of nuclear speckles, these fixed points, the DNA locus. And the same for the heterochromatic regions, as well as the heterochromatic locus. So the green dots is basically caked on top of these green blobs, the heterochromatic blobs. And same for nucleolus as well. And this is true actually across many different cells. So, we're gonna, here's another cell, and same for the speckles and heterochromatin. You can see again, they're caked on the surface. And I should mention these fixed locus is generated across all the cells, right? This is bulk averaged. So you can see some of them do deviate from being associated with the speckles, but most of the cases they are attached in single cells, and they're on the exterior, living on this two-dimensional surface rather than fully space filling in the nucleus. And this is also true for other chromatin marks. You can see for the acylation mark as well as this heterochromatic mark. Again, they live on the surface of these nuclear blobs. Just another cell to show you that you can see these features. You can look at all the cells and all basically show very similar features. And so now if we zoom into individual chromosomes, so in this part of the cell, you can see there's chromosome 4. And on average, in the bulk measurements, we see that the chromosome 4 have parts are attached to the heterochromatic regions consistently in the beginning, and then parts are attached to the speckles at the end of the chromosome. And you see in the cell, right, that that's what you see, that this is the beginning part of the chromosome, this is the end of the chromosome, in the middle there's going around. And this is features you see again in many different cells. So in chromosome 4, you can see, in this cell, there's two alleles, and they have this attachment pattern. And if you look at chromosome 17, it has a different pattern here, but again, in single cells, they're consistently attached to different parts of the nuclear bodies. Same for the nuclear, different chromosomes that attach to different nucleolus, as well as the nuclear speckles. So the picture for us is this, that even though the individual chromosomes in individual cells are highly heterogeneous and variable, in fact, their structures are very deterministic because they're anchored to these nuclear bodies in individual cells in very consistent fashion. And so that suggests the nuclear structure is actually very, very deterministic and invariant. And this is very important, I think, because in physics, there's a one-to-one relationship between invariance, things are invariant, and also actual physical equations by consequence of Noether's theorem. And I think in biology, because we don't have equations, the things that are invariant are actually very important because of the best we can do in some sense. And so, on different chromosomes, across all 20 chromosomes, you can, generate these fixed points. And then this gives you like overall scaffold of how the chromosome is organized. And so the last thing I'm going to mention is that on the single pixel level, you can you can cluster based on the combinatorial profiles of all of these antibody marks, and you can generate these zones that tells you what the combinatorial features of each one of the regions nucleus are. And when you put the DNA on top of that, you see that actually, the DNA exists mostly on the interfaces between these distinct zones. And this is really clear if you look at the active genes. So, we look at the active genes by targeting intron, which tells you the genes are bursting at that given time. And you can see they basically live very much on the interfaces. And if you look at what the RNA polymerase, active RNA polymerase marks are, you zoom in, you see that the introns are actually living not in the inside of these RNA polymerase condensates, but actually on the edges of RNA polymerase. And this is actually very surprising. We don't know why this is. This is something to be looked into much more further. Okay, so let me just wrap up and say that, I think the takeaway message is that the nucleus, the chromosomes are not these sort of spaghetti ball things that are very compact. organized, but rather they're almost like inside a baguette. There are a lot of holes in the nucleus, and the DNA is actually living on these pseudo two-dimensional surfaces, and it could be interesting implications of how things are regulated. So yeah, finally, just let me thank my group who who really built up this sequence technology in the last decade or so, and particularly Udai, who was a master of DNA fish and integrating all these methods together. And thank you very much.
Thank you, Long, for the talk. I had trouble to unmute myself because I was so immersed and fascinated that I just couldn't find the mouse anymore. I'm sure I'm just one of hundreds with that impression. So questions will be in the comment session. So now I will need to introduce Felix Hartmann, who also was or still is in Stanford, but who is now joining as a group leader the German Cancer Center. So that's exciting also. And Felix, the floor is yours.
Well, thanks so much for the introduction. Let me see if I can get the screen sharing going. All right. Yes, okay. Well, yeah, again, thank you very much for this introduction. And of course, also for inviting me to this workshop, which is really exciting. So I want to talk to you a little bit about a project that I've been working on. And really I would say in a broader scheme, my research involves like these two technologies to measure protein expression patterns, both in single cells, but also using a multiplexed imaging platform to look at this in a spatial context. And I've really also been sort of working on developing new tools to look at, I would say, aspects of cellular biology that we maybe often overlook simply because we don't have the assays to study them easily. And I think these can be very important, of course. And I think that, for example, cellular metabolism is really a prime example of this. So I became really interested in cellular metabolism sort of in the context of cancer immunotherapy, which is what I'd been working on before. And it turns out that on the one hand, we know since a long time that, for example, tumor cells are metabolically very different. But more recently, maybe we've learned that this is also true for many other cell types, including these immune cells, where really cellular metabolism has been found to be very important in driving various of their functions. So for example, the production of effector cytokines, whether these cells differentiate, for example, T cells into effector or into memory cells, and also, for example, whether these cells become exhausted. So really important functions are determined or influenced by cellular metabolism. And then on the other hand, if you think about the tumors, they are also known to actually metabolically influence the cells around them in the tumor microenvironment, including these immune cells. And they can use this sort of metabolic influence to induce, for example, immune suppression, dysfunctional phenotypes, and so on. And that, of course, affects outcome in the end. Now, if you think about how how this actually looks, of course, in a human tumor, it's a very complex interplay. You have many different cell types in the tumor, including all the different immune cells, but you also have stromal cells, such as fibroblasts, you have the vasculature, and then you have the tumor cells, which can also be very heterogeneous. So really, if you want to study these metabolic interactions between these different cell types and how they influence each other, you really need a technology that is that can look at this on a single cell level, and of course, ideally also with spatial resolution, so you can figure out which cell types interact here. So really, in order to address this, I've been working on this approach, which we call now single cell metabolic regulome profiling, or CMEP for short. And sort of the underlying idea here is that what we want to look at is the so-called cellular metabolic regulome. Now, what do I mean by that? So if you take a given metabolic pathway, I want to look at all the things that really determine the metabolic flux through this pathway. So not necessarily the metabolites themselves, but everything that determines flux, such as, for example, metabolite transporters that take these nutrients and import them into the cell, rate-limiting metabolic enzymes that catalyze these reactions. We can also look at post-translational modifications of some of these proteins to get an idea about the actual level of activity. And then, of course, there's signaling molecules in the cells that can react to certain metabolic states and transcription factors that we know drive sort of metabolic remodeling. So all of these things, all of these components determine together the metabolic flux, and all of these we can actually detect using specific antibodies on the protein level. And in our case, these antibodies are coupled to heavy metal isotopes, and so we can quantify them using these two technologies. One is single cell mass cytometry, or CyTOF, and the other you heard about already, I think, is multiplexed ion beam imaging, or MIBI. So I think most of you have heard by now of mass cytometry, or CyTOF, so I'm not going to go really into detail. It's a single cell technology that allows you to study, as I said, protein expression patterns via mass spec quantification of these antibodies that have the heavy metal isotopes on them. And the really related technology is where you can do imaging, because you do this on a pixel by pixel level. And so you get these high dimensional images, is multiplex ion beam imaging, or MIBI. And as I said, I don't want to go into the details of how it works, but I think these technologies are really nice because not only can you look at many parameters at the same time, sort of 40 to maybe 70, they're also really, really sensitive. So in some cases, depends on the exact channel, but you can get down to single molecule sensitivity in order to detect this. And I think another really important point is that they're really fast. So you can do, with CyTOF, you can do millions of cells a day, or you can look at hundreds of fields of view, for example, with the imaging platform. So really allowing us to look at this in human clinical cohorts, for example. So the first thing we did in order to validate this metabolic approach was really to take human T cells, and we stained here for some glycolytic regulators. And if you do this, for example, resting T cells, you will see that they're, they don't need much energy. They're not doing much. So for example, they have very low levels of the glucose importer and an important glycolytic enzyme hexokinase 2 here. But then upon activation, these cells need to proliferate, produce cytokines, and so on. So there's really a drastic upregulation of these glycolytic regulators. And so we can use this data to compare this to sort of a more traditional metabolic method measurement, which is called CHOR, so extracellular flux analysis. And so what we did here, we really integrated information from all of these different metabolic regulators into a single glycolytic score, as we call it here. And then we can compare this with the actual level of glycolytic activity here. And if we do this for this T cell activation time course, we find that the two really agree well. So, and the same was actually also true if you look at things that are related to oxidative phosphorylation. So that was, of course, exciting because now we could go and really use this approach and answer some questions that were hard to get to before. So for example, we took a small cohort of human colorectal carcinoma patients and controls. And again, you can combine this, of course, easily with looking at surface markers. So you can identify really all the different cell types in these tumors here. I'm going to focus on CD8 T cells. And first of all, what we saw is if we now only look at their metabolic features, is that these are very heterogeneous. So you find multiple metabolic states really of these T cells. And you can, of course, then look, which different pathways are switched on and what are they sort of characterized by. But what I want to point out here is just one little thing. For example, if we looked at which cell types are found in which tissues or which metabolic states, we found that there was this one state 3, which is shown in red here, that seemed to be really specific for colorectal carcinoma and that we didn't really find in any of the other tissues. And then, as I said, we had defined these cells metabolically, but we can go and look at their phenotype or at their function. And we see, for example, these are sort of non-proliferating, TCF1 low, but they do express or they are enriched in CD39 and PD1. So that was, of course, very interesting because these markers are often used to denote exhausted cells. But we here sort of metabolically defined these cells. So I'm really excited to see, for example, if this might be a useful definition of these cells that can go beyond these surface markers, which can also be expressed by, for example, recently activated cells. So that, I think, overall just shows us it's like a little proof of concept study that we can use this approach to do metabolic profiling directly in clinical cohorts. And that this metabolic profiling actually identifies functionally and also clinically relevant cell states. So now I want to switch to the imaging. And as I said, we use multiplex ion beam imaging, which you can directly apply to human tissues. Again, I chose here colorectal carcinoma. So take FFPE tissue, and I stain it with a combination of course, immune targets, so we can identify all the different cell types, and then also metabolic targets. And it's flipping through all of the 36 or so channels here. But of course, we can not only look at these things in isolation, we can do things like multicolor overlays. So you can now really start to figure out sort of the spatial arrangement maybe of these metabolic phenotypes in these tissues and how they're arranged. So one thing we did here, if you remember in the beginning, we integrated, for example, information from all of the glycolytic regulators, and we can do the same on the imaging platform. Now we can get basically a glycolytic score for every single pixel in these images. And what is shown here is sort of, these are, for example, 2 different immune cell infiltrates within the same patient, within the same tumor. You can see that this would suggest that they have very different glycolytic activities, which we can now also think about why this is and what does that mean for this patient, for example. Beyond that, we can, of course, as multiple people have already indicated, we can do single cell segmentation from this data, which we did here. And then we can look in the local neighborhoods, around any given cell. What is the metabolic phenotype here? And what we see, for example, if you look at cells that express, let's say, a high level of a glucose importer, GLUT1, if you look in their little microenvironment, you'll find other cells that are also expressing high levels of this glucose importer. And not only that, you'll also find other cells that express other glycolytic enzymes here. And this was true really for multiple metabolic pathways, such as oxidative phosphorylation, amino acid metabolism. So there seem to be these sort of local niches, if I can call it like that, where cells really sort of, where cells are maybe driven to use a particular metabolic pathway. And I think it's going to be interesting to figure out why that is. Beyond that, we can also do it sort of the other way around, and we can identify structures in these images and ask how do they influence cell metabolism. So this is an example of, again, colorectal carcinoma, where you see the tumor cells here in magenta, and the immune cells are up here. And what we did here, we defined a tumor immune border. And again, we took the immune cells that are sort of close to the tumor in this white band here, and we metabolically compared them to immune cells that are further away from the tumor. And if you do that, what we saw is actually there seems to be a broad polarization, metabolic polarization of these immune cells towards the tumor. So they've increased the expression of a lot of different metabolic enzymes. And I would say a really interesting was, for example, upregulation of amino acid transporters towards this tumor immune border. So again, I also think it's an example of how we can use this technology to understand, how does the metabolic phenotype and the tissue structure, how do they come really together and influence immune function, and at the end, of course, also sort of clinical outcomes. Yes, so just to sum up what I've told you a little bit, so I think this CMAP approach really provides us with a novel lens to study cell metabolism, in addition to these other maybe bulk methods that are already out there. This one has, of course, single cell resolution, which allows us to study heterogeneous samples with different cell types, maybe also rare cell types where you would have problems to isolate many cells. And of course, the other interesting thing is multiple populations at the same time and seeing how they maybe influence each other. We can also, and I think this is very interesting from a multi-omic perspective, we can combine these metabolic measurements with measurements of cell function or phenotype or other things using antibodies. And of course, we can look at the spatial arrangements of these metabolic programs directly in these human tissues. So together, I think that this is really going to enable us to do now really translational studies in human clinical cohorts, see really how these things are changing in, for example, in cancer patients upon response to treatment, and how these things can they be used maybe as targets for future therapies, or can they be used to predict things? So all of these questions, I think I'm excited to address in the future. So we've started already some of these projects, really all over, not limited to tumor immunology, but various different topics ranging from stem cells, to cancer, to influence of diet, for example. And I'm sort of really excited to see how would these metabolic programs, how do they influence outcome in these different diseases and these different projects here. And sort of, I think, beyond applying this and looking at this in a clinical context, I'm excited to sort of further develop these approaches And one thing I've been thinking about recently is, so I started this talk basically by saying that, cell metabolism influences immune cell function. And if you think about how does it do that, well, there's multiple avenues actually how it can do this, but one very prominent way is by influencing sort of epigenetic remodeling or epigenetic imprinting. So you can imagine that, in order to, for example, do histone methylation or acetylation, well, you need the substrates. You need, for example, acyl-CoA for acetylation and SAM for methylation. And these are, of course, provided through metabolic pathways. So I'm really excited to look at this connection with the energy metabolism and how they can provide these substrates, but then also at the same time, really look at the levels of these epigenetic marks, which you can also, for example, assess with antibodies. So integrating these two measurements, I think, is going to be really exciting. So with that, yeah, I want to thank everyone that was involved in this project, Sean, a mentor, and also Mike, our collaborators from Amsterdam and from Caltech, sponsors, and as Nikolaus already said, so I'm excited to do the next steps, and I will be opening a lab in Germany soon. So if anyone is interested in using these approaches or developing some new ones, just shoot me an email. And yes, thanks everyone for the attention.
Thank you, Felix, and also perfectly on time with a great talk. So, and now we come to the joint Q&A. And I think what I'll do is I'll just pick some of the questions, and I'll not necessarily keep the order of the speakers, but just ask some questions which are maybe of interest. And I'll start with a question from Robert Zinsen, actually a colleague here in the Institute, goes to Long. And the question is genome topology more stable along lineage descent or cell type?
Yeah, that's a great question. We haven't looked at cell type so much, but we looked at, in the inside of a colony of cells, how much are sister cells and cousins related in terms of structure of the chromosomes. And so sisters are very correlated in terms of the chromosome arrangements. But one more generation later, they're essentially completely uncorrelated. So I think there's a very simple explanation for this is that, when you go through mitotic division, you have to line up the chromosomes, right? And then you separate them. And so in two daughter cells are, the structures are roughly correlated. And I think that's all we see. Like after one generation, it's all gone.
Mm-hmm. I mean, along these lines, people are discussing a lot about condensates, right? And so this idea that there's a maybe dynamic condensation happening in for example, in the nucleus, are maybe your data compatible with this? Would you say, and that the fixed points then seem to be a kind of the stable anchors that pin the structures to some global overall stability?
Yeah, I mean, I think this is also getting a chicken egg question, right? Like,
sure.
Which comes first, the condensate or the DNA locus? You can see in those single cells that if you have the fixed points for speckles, they don't all have to attach to the same speckle. They could, like in the same chromosome, they could attach to different speckles. So, there's a lot you need to do, I think, to do some live cell experiments to get at that. How does the condensate form? But I think this also suggests that condensates are excluding DNA in many cases, and the DNA is not inside these protein blobs. But I don't think we know too much about them yet.
There's a question for John, a technical, straightforward question perhaps. How efficient was your cell segmentation? What percentage of cells are accurately segmented? Was this a challenge? Comes from David Granger.
The short answer is yes, it is a challenge. I think that the membrane stain that Tim worked on together with colleagues in Long's lab was absolutely essential, these 4 additional membrane markers. Without that, I think you could do, nuclear segmentation is relatively straightforward, but then you just expand from a nucleus out, and drawing the margins of a cell is extremely hard. We were actually looking, I was just looking earlier today at some of the earlier time points, and there the degree of the density of cells is so tight that the membrane markers there are quite difficult even to use for the segmentation. So there it might even be the manual segmentation to an extent becomes more effective than trying to train something computationally at the moment. So I think it depends a little bit on the type of data you're looking at and how dense the cells are. If you have a good membrane marker, you can train a machine learning approach and test its performance pretty well. And Elastic in our hands worked really quite nicely for that task. I think without a good membrane marker, then it becomes much more difficult because you're playing with various watershed algorithms and other types of approaches in order to do the segmentation. And the lack of a gold standard can make it pretty difficult. I mean, Long has looked at a lot of these images, so he's probably able to comment on how easy it is to segment seqfish data more generally, but I think it's not a trivial challenge, right?
Long, you want to comment on this? Yeah, that would be great.
Yeah, I mean, segmentation is actually usually the hardest part of data analysis. So it is still a major challenge. Elastic helps a lot, but there's still situations where it's very difficult to train something and to segment it well.
Okay, so there's another technical question for John, which is about it's by Jan Lause, and I'll just maybe read the last sentence of this question, which is, how do you handle cells that do not find a close neighbor? So,
right. So there are different ways of doing that. So first, we don't just, we take a consensus approach to basically when you map the two data sets together, you look at the neighborhood of cells in the genomics data and take a consensus view of the neighbors to assign a cell an identity in the seqfish data. But there will be some cells that either due to embryo preparation where maybe a particular region was dissected out before, say, the sequencing experiment, and that's present in the seqfish or vice versa, where assigning a good identity is hard. And in principle, the nearest neighbors approach that Lally developed should keep those quite separate, and they'd have a low confidence of being near another cell. So they've just basically assigned a non-identity or unknown identity. And that's important because otherwise, I think it's a challenge of some of these integration approaches that they sometimes have a tendency to slightly over-merge data sets, and you can end up erroneously mapping cells on top of one another. And it's particularly a challenge when you've got, say, a disease in a normal cohort, and it's a subtle effect in terms of cell type identity. Then the mapping of the two data sets on top of one another can be hard. So you want to assign some almost an unknown or unidentifiable label. So we would do that.
I think this was very diverse techniques and talks, of course. So the questions also reflect this. I wanted to read a question for Felix, but as I'm reading it, it disappeared from the Q&A. But no, there it is. So the question comes from Ken Chen, and the question is, are there metabolites that cannot be sufficiently observed through antibody staining? I think you didn't really talk about metabolites, and that is indeed a challenge, but I think the question is good in the sense of that you can maybe talk about the perspectives for really looking at metabolites.
Right, yes, exactly. So just to say this again, we're looking basically at all of the adjacent proteins that would sort of catalyze these reactions and determine hopefully how much metabolite there is. Although I wouldn't actually go so far as to say, oh, there's a lot of protein X which catalyzes this reaction, so there has to be a lot of the immediate substrate there. I think of it more as we measure, let's say, 5 or 6 glycolytic regulators, and we get an idea about the glycolytic activity of the cell, sort of the overall flux through this pathway. But yeah, metabolite measurements themselves are tricky. Actually, a lot of metabolites are, first of all, very small, so they're hard to get at with probes. Secondly, they often have super short half-lives. Sometimes this can be, seconds to minutes. So it's really hard to do an assay. You can imagine if you isolate cells or if you sort them or anything, a lot of metabolites can disappear. But that being said, there are some that can stick around. I think we are getting quite good, for example, in using sort of mass spec de novo imaging approaches to look at some of these metabolite distributions, maybe lipids, I think is an interesting category there. And I'm excited to see how these levels overlay. I think there's going to be differences, for example, in how, how sensitive these methods are, how the resolution of these methods is very different. But I think in general, I'm excited to look with some more traditional metabolomics methods and how can we integrate these data sets. I mean, if we go away sort of from the single cell level and we think about, you can measure lots of cellular metabolites in sort of bulk and try to correlate that back with other measurements and maybe try to integrate that with the single cell data, I think that's going to be also very interesting. Yeah.
Okay. There's another question for Long, which I will be reading from David Granger. Did you observe missing signal for highly compacted chromatin regions?
Well, I think not really. There's not so much a bias against highly compacted regions because you can target these against these satellite repeats, which are in these heterochromatic, heavily repressed regions, and you can detect them just fine. But definitely, the detection efficiency for DNA is not 100%. It's we estimated about 50% of the locus are detected. So some things are. Some locus are definitely missing, and we're not quite sure exactly why they are.
Mm-hmm. Okay, so I'll do now something maybe unconventional, which is I'll ask the chair of the first session, Fabian, to maybe comment if, based on what we had in the first session, you see connections or things that would come together here with the second session.
Yeah, I think this is totally disconnected. I was quite impressed, and I actually didn't expect. Maybe I should have, but Long really being like the whole subcellular part. I wonder, maybe just like sort of as a provocative thing, because then Felix, right? So you put them then these more complex tissues, and then done also the whole development. Do you guys think it's time to take up this whole idea of multi-scale? I don't know if it's modeling right away or analysis, something like that again, right? I mean, all of this stuff obviously is connected, and I guess a little bit with when we had this disassociated stuff, right? We've been now starting to connect with cell interactions to sort of one level higher. That was a bit of a theme of the first discussion, right? And now I guess you could ask similarly, maybe if Long really resolves certain variation of intracellular things, right? Also spatially positioned, could we connect that one level higher somehow? Cross scales?
Yeah, I mean, it's definitely something very interesting to think about. I mean, I think the short answer is absolutely, yeah. Definitely should connect them. The DNA data I would say right now is mostly megabase, so it's a little hard to say something about regulation because the 1 megabase region has many genes. But the one thing I didn't talk about was how actually if you just look at the immunofluorescence markers, a lot of them are very heterogeneous in different cells. So for example, H3K9 acetylation is, if you just look at the global level, is not homogeneous across all the ES cells. And they actually don't correlate completely with the RNA signatures. So, in ES cells are supposed to be in high and low states, and some of these chromatin mark variations actually, don't match with that. So there's something happening in addition to the RNA level. And so, I have to think more about the multi-scale integration thing, but definitely the multimodal integration, just the fact that you're measuring more things.
That's the more obvious part, you're right, yeah. So basically just adding essentially delocalized stuff. Okay, that makes sense. And but you say there is something where the integration should not be as straightforward because you see something that you don't essentially observe in dislocalized attack or also within chromatin or methylation.
Yeah, I mean, well, we don't quite have the resolution to see the individual genes, right? So that makes the integration a little harder in that respect. But I mean, I think this is very thought-provoking, and I just need to think more about it.
I think now I'll say the following, and there are supposedly closing remarks, which I will not really do. I think we can do this together if you want. But I think it is clear that the description, the spatial molecular description in space is making enormous progress. If I look at today's workshop on all levels, right? And I think this will continue. There's no question about it. And so we will get all these data in space from different levels. So the question is, what do we really do with them? And I think John made, to me, a very important point about that. It must be also about understanding really the mechanisms behind this, which organizes and what is the function of this kind of organization. Let's say Long's data really scream for, trying to understand why this is and why are things organized in the way we see it. There is an obvious immediate application, in my opinion, from these descriptive data to disease, which is what Felix was maybe the closest in pointing out. You can already take these data, and of course, from the first part of the workshop also, we heard a wonderful example of triple-negative breast cancer. So there's an obvious clinical application, so you can classify patients better or diseases better, and maybe even predict drug targets in space. I think that is also to come. But having said all this, I think the grand challenge is really to understand, and so there we need to perturb, and so to bring up this point again, I think it will be key to develop also methods which allow, as already a little bit discussed with John, let's say surgical spatial interference, or interfering in a spatially programmed way, let's say. I think to do this with traditional genetics is incredibly difficult to think about, and so probably new approaches are being developed or will be developed. I think it's just very important, and for future workshops or so, this is maybe also interesting to discuss further. But now I want to give the floor over to you. To maybe make some final words that you would like to respond or add.
So I assume you mean the three of us, Long, Felix, and myself, are. So I would agree that and I think it's really exciting because there's a long and fairly extensive literature on, say, patterns of morphogenesis, on diffusion gradients, and based around, say, different differential equation models for how things have developed. And we are getting close to an era where we're going to have, at the molecular and spatial level, enough information to begin to integrate those different types of models. And that will allow some principles that have been proposed and maybe sometimes not experimentally validated, at least not at the systems level, to be properly tested. I think we have to think very hard about doing the right perturbation. I agree with you that a gross perturbation is not going to be very informative. So the right perturbation, so in a lineage progenitor, and in a way that's going to be subtle enough for nothing to be too gross, but also meaningful enough that we could test a hypothesis, is going to be really important. I think it's really exciting that this bridging of theory together with 4D organismal biology is going to be a really exciting feature of our scientific discussion over the next decade. And that's a really cool place to be, both in the context of normal development and a disease. So I'm really excited about this. It's going to be a great decade once COVID is done.
Yeah, and I would just agree with John that I think development is really, something that I think we're super interesting that we can start really to address with all the spatial tools and the integrated tools, right? Like you can measure all of these things at the same time, and then plus lineage, which we didn't really talk about. There's all these tools to track lineages and to integrate all of these things together. You can generate a complete map of development.
Yeah, I mean, there are, before Felix or Fabian comment, I mean, there is, of course, the one problem, which is a developmental problem, okay, and then there's the other problem, which is a homeostasis problem. So both things are extremely important. but they are a little bit maybe conceptually different. And although the mechanisms behind will be related, but that is still something to keep in mind. Felix and Fabian, your last comment on.
Yeah, no, I think, I mean, I agree, of course, with a lot of what has been said. I mean, I think when it comes to perturbations, I have also, of course, been thinking about this in sort of a clinical context on the patient level, actually. And, you know, if you think about drugs that can maybe, such as, I mean, I think I'm going to look at a project looking at VEGF inhibitors, so where we can maybe, you know, remodel the vasculature in a tumor that has an effect on nutrient supplies, and I'm hoping to use sort of these sort of inpatient perturbations to find out some of these things and how does it actually, you know, look when you give a patient a drug. But on a more general note, I think, yeah, what others said, I'm really also super excited about taking these approaches and sort of applying it more clinically and to patients and integrating multiple measurements that we're sort of very good at doing sort of in a cell biology level and on bulk, but not so much maybe yet in clinical contexts.
Fabian.
I'll be fast. You might hear there's a kids party already starting at my house. So, you know, Germany kind of getting done. I just have this one thing, spatial context, right? So we're still having this genomics hat on. It's essentially cells with XY coordinates, but you know, obviously there's like a bit more, right? And I think for me, maybe nowadays just trying to build the infrastructure that we can sort of put tools on top of it for the analysis of getting things together. At the moment, on things like RNA-seq, it was very easy to take stuff that John was doing out of the box and apply to our stuff. For the spatial things, it's not that anymore. So I think like sort of agreeing on a bit more standards there and then sort of having things to exchange, I think could be a fun starting point so that there's this whole rich community happening, and that could be fun.
Super. So we're just 3 minutes over time, and we had a joint closing statement that was really fun. I think I want to say on behalf of all the participants, thank you to the speakers, and enjoy your evening. Ciao.